And thanks for inviting me. So I'm going to talk about work that has been going on in the lab for the last 5 years at least. And I've been involved in the last part of this project, and especially for the agent-based model part of the story. And the story is about modeling the immune response to tuberculosis infection. So let me give you some facts about TB. It is a deadly infectious disease caused by the Mycobacterium tuberculosis, and 1/3 of the world population is estimated to carry the latent infection. We're talking about 2 billion people. The current vaccine is not really reliable. It works in children somehow, but it's not really very effective in adults. And there is a treatment. Although approximately 2 million people still die each year. To make it a little bit more dramatic, someone in the world is newly infected with TB pretty much every second. And 35 people die of TB every 10 minutes. So that's a big problem. The bacteria is a pathogenic species. It's been discovered by Robert Koch in 1882. He won the Nobel Prize for that. It's a gram-positive bacteria, highly aerobic, mostly intracellular, and one of its virulence factor, although it's not classified as virulent factor, is its very slow growth, which makes him sometimes undetectable by the immune system. So it's kind of an evasion mechanism by the bacteria, which has been involved for many, many years, thousands of years. So a little bit about the epidemiology. There is a chance for clear the infection upon exposure, but that's not really clear. So we think that the bacteria is so successful that almost 100% of the time can colonize the host. But those data are not really clear. What we know is that from 5 to 10% of the people that have been exposed, 5 to 10% progress to primary TB, which means that if it is not treated, the host dies. The majority, however, of the individuals that are exposed progress to a latent infection called latent TB. It's asymptomatic, and the host can carry it for its lifetime. And this is a large pool of individuals. I'm talking about 2 billion people. What happens over time that due to other factors such as colonization, co-infection, HIV infection, you can experience reactivation. As a matter of fact, the majority of the major cause of death for HIV people is TB. So what happens is basically the host reactivate the infection, and it goes back to the active primary state. Now, we do have 9 million new cases per year, so that's a big problem. On top of that, there is a treatment, but non-compliance is an issue because it's a lengthy treatment. And so we end up with drug-resistant TB, which is a major problem worldwide. So it needs longer treatment, and sometimes there's no treatment at all. So what do we do? Well, first thing I want to talk about is try to describe very generally the course of infection, and then how we deal with that and try to model it. Rob gave some ideas on trafficking of cells from the site of infection to the lymph node and back. This is the same thing for TB. It's a part of the adaptive immunity mechanism. So you got an infection in the lung, you have dendritic cell and macrophages as well as neutrophils being recruited to the site. Macrophages become infected and are less, let's say, more permissive, if you like, than dendritic cells. They experience, infection, while dendritic cells, upon infection, they start to mature and they express, a different phenotype. They become migratory, and basically they migrate through the closest draining lymph node, where they co-localize in the T cell zone and they prime naive CD4 and naive CD8. So in the lymph node, basically there is this effector T cell generation, and cells that are generated there that migrate back to the site of infection to do what they're supposed to do, The first step and the necessary step is macrophage activation, which means the macrophage that is infected, it's activated so it can kill the intracellular bacteria. Then there are the mechanism, which is CTL killing, for example, or apoptosis induced by TNF or fast ligand, other mechanism related to T cell. In the midst of all this, the hallmark of TB infection is the granuloma. So we have timing and different cells that together form this cluster, organized cluster of cells, and that's called granuloma, and usually forms in the lung. The idea is that the granuloma can control the replication. It's kind of walling off the infection, so it's a defensive mechanism by the host. And let me see. All right. So if this works, typically we get these granulomas that they have some fibrosis inside, and which is linked a little bit to the fibroblasts that we were talking about in the previous talk. And so at this point there is kind of a healing mechanism, and the granuloma can heal. There are some cavities, but the bugs is gone. If there is an inadequate control of replication, basically we progress to active TB, and the granuloma is a little bit disorganized, and the bugs, those red dots, can get out and can disseminate through the blood to other organs. Usually secondary lesions are not in the lung, but are in other organs. So we want to look at granuloma formation. More generally, we want to look at the whole system, which is a complex system. Here we just superimpose different layers for different organs. On the left, you see it's the lung. On the right, it's the lymph node. And you can zoom in and zoom out at different levels, at different scales. At the tissue scale, you look at the granuloma. You zoom in, you look at the cellular scale, cells comprising the granuloma. You can zoom in even further and look at receptor ligand. The same thing can be done on the lymph node. So when you look at the whole structure, you zoom in at the T cell zone and look at antigen-presenting cell T cell interaction, and even further the molecular scale MHC complex dynamic, peptide MHC complex dynamics. Answering to Heber, where do we even start? Well, the claim could be, well, it doesn't matter where you start. In the best of all systems biological world, we're going to meet somewhere. And so you can start from, if you're good in, molecular scale dynamics, you can start from here. In TB, what's really important is granuloma formation. So we decided to start from there. Let me tell you a little bit how we, proceeded in this analysis. Our middle-out approach is first to look at these different scales and try to see where, the relevance of events at different scales. So one scale can be observed and impact a different scale. That could be higher scale or lower scale. We can look at different organs, and the other idea is tunable resolution. We can zoom in and zoom out. So if really, depending on the question we're asking. The granuloma is definitely a multi-scale problem, and that's both in space and time. Now, we do have data, not a lot of data, and there are several established animal models for TB and granuloma in TB. There are limitations. The most common is the mouse model, but there are limitations in terms of lack of latent infection, so it's hard to replicate human infection. Infection is chronic and progressive. The pathology of the granuloma is different. The best model is the human primate model, but that's expensive. And given the slow-growing nature of the pathogen, it basically, those primates are in cages for years. I mean, if you really want to replicate latent infection, and that's expensive, and it's hard sometimes also to decide when to sacrifice